Welcome back to the Deep Dive. Today we are taking a massive run at a cellular landscape that is, it's far more complex and dynamic than any city grid you could imagine. We're talking about the cytoplasmic membrane systems.
Right. And if you've ever stared at a cell diagram, just trying to figure out how a protein gets from point A to point B, like from the nucleus to the cell surface.
Yeah, how does it know where to go?
Exactly. This deep dive is basically your comprehensive shortcut to understanding that.
Our mission today is to guide you, the learner, through, well, every major concept, process, and even the key experiments in this chapter. We want to make sense of the endomembrane system for you.
That's right. We're focusing on that interconnected flow between the ER, the Golgi Complex, endosomes, lysosomes, all of it. Think of it as mastering the internal logistics network of the cell.
And we're starting with a massive hook, a real-world application that is, frankly, a battle for survival. The idea of hijacking the cell. When you look at the opening micrograph in the chapter, you see HIV, the human immunodeficiency virus, clinging to a host cell. And it's just this striking reminder that survival, whether it's for the cell or for an invading virus, comes down to mastering these membranes.
It really is a battle of wits at the nanoscale. I mean, for a virus to get in, replicate, and then get back out, it has to conquer these internal systems. It can't just, walk past the checkpoints.
So, okay, how do they get past that first barrier, the plasma membrane?
Well, the strategies are varied and honestly pretty elegant. Some viruses, like herpes, they just go for the direct approach. They fuse straight with the plasma membrane and dump their genetic material right into the cytoplasm.
Simple and effective.
But a much larger group, they're stealthier. They hijack the cell's own internal pathways, the endocytic pathways, so that they basically trick the cell into swallowing them.
So they get pulled inside, hidden.
Exactly. They're hidden inside these protective bubbles called endosomes, and they only unleash their genome once they're deep inside the cell. It minimizes any traces on the surface that might, trigger an immune response.
And once they're in, they need a factory to replicate, right?
Yeah.
And viruses, being the ultimate parasites, they're not building their own.
No, they're remodeling. They're fundamentally restructuring the existing cellular architecture. It's an incredible process.
So they find the existing machinery and just take it over.
They do. Researchers have found that viruses carry out this really elaborate restructuring of the host cell's internal membranes, especially the endoplasmic reticulum, to create these specialized, almost organelle-like structures. They call them virus factories or viroplasm.
Wow, so the cell's own sophisticated assembly line gets completely co-opted.
Precisely. Inside these virus factories, you have viral proteins organized into these dedicated assembly lines. It's like they've built their own scaled-down specialized organelle just for mass-producing more viruses. It's a stark contrast to the thousands of different products the normal ER and Golgi are juggling.
It really hammers home how important that compartmentalization is.
Yeah.
Even a virus gets that structure is essential for function.
Right. And that idea of compartmentalization leads us to the big picture overview for this chapter. When scientists first got their hands on electron microscopes in the mid-20th century, they had this huge revelation.
That the cytoplasm wasn't just soup.
Exactly. It wasn't just an open homogeneous space. It was this incredibly subdivided city of membranes, a dynamic integrated network.
It was a revolution in how we saw the cell.
Absolutely. We realized the cell's internal space is broken into these distinct compartments like rooms in a house, if you want an analogy, and each one has a unique set of proteins and a very specialized job.
And if you look at a high-res image, say, of a maize root cell, the sheer surface area of these internal membranes, the ER, the Golgi, it's many, many times greater than the external plasma membrane itself.
It's vast. And these components, the ER, Golgi, endosomes, lysosomes, and vacuoles, they form this one dynamic, integrated endomembrane system.
Now, a crucial distinction before we dive in. Mitochondria and chloroplasts are not part of this system, right?
Correct. They have their own origins, their own import systems, though interestingly, other organelles, like peroxisomes, seem to have a dual origin. Their boundary membrane can arise from the ER, but most of their proteins are imported later from the cytosols. Complicated.
It always is.
Yeah.
Okay, let's get into the mechanics.
Right.
How does stuff actually get moved around inside this system?
The primary method, sort of the main highway system, is vesicle transport. Materials shuttle between organelles in these small, membrane-bounded transport vesicles.
And this isn't just random floating around. It's highly directional, like delivery trucks on a tight schedule.
Oh, it's highly organized. So a transport vesicle will literally bud off from a donor compartment. And here's the key part. As it's budding, it's already incorporating specific membrane proteins and its soluble cargo, which is often bound to receptors on the inside.
So it's prepackaged with exactly what it needs.
Exactly. Then this vesicle gets pulled through the cytoplasm, usually by motor proteins that are running along these tracks made of the cytoskeleton.
Microtubules and microfilaments.
Yep. And when it reaches its destination, the acceptor compartment, the vesicle fuses with it. And in that fusion event, it transfers its soluble cargo into the lumen, and just as importantly, it integrates its own membrane wrapper into the acceptor membrane.
So if vesicles are the main highways, does the cell ever use, I don't know, shortcuts? Are there other ways to move materials?
That's a great question, and it brings up a really fascinating and relatively new insight: membrane contact sites, or MTS.
Okay, what are those?
These are not fusion events. They're these specialized regions where two organelles, say the ER and a mitochondrion, are tethered together in extremely close proximity. We're talking maybe 30 nanometers apart, but their membranes never actually merge.
What's the point of just getting close?
It allows for the direct, highly regulated transfer of small molecules, especially lipids. It's also vital for signaling between those compartments. It's a very localized and energy-efficient transfer route that completely bypasses all that complex machinery of vesicle budding and fusion.
Interesting. Okay, so let's define the two major traffic flows in this cellular city. You mentioned outgoing and incoming.
Right, the two major pathways. First, you have the biosynthetic secretory pathway. This is the forward flow, what we call anterograde transport. Proteins are made in the ER, they get modified in the Golgi, and then they're dispatched to their final destinations, lysosomes, the plasma membrane, or maybe vacuoles in a plant cell.
And when that material is meant to actually leave the cell, we see two different ways of doing that final discharge, right?
Exactly. There's constitutive secretion, which is just this continuous, unregulated stream. Most cells use this all the time to renew their plasma membrane or to build the extracellular matrix around them.
And then there's the other kind.
Then there's regulated secretion, which is highly specialized. Here, materials are synthesized and then stored in these large, densely packed secretory granules, and they're only discharged when the cell gets a specific signal.
Like insulin release when blood sugar goes up.
The perfect example. Or a neurotransmitter being released when a nerve impulse arrives. It's secretion on demand.
And operating in the opposite direction, you have the endocytic pathway, the retrograde flow.
Right. This moves materials from the cell's exterior surface inward through compartments like endosomes, and usually ending up at the lysosomes, where things are typically broken down and recycled.
So all of this complex targeted movement has to rely on some kind of internal mailing system. This is protein targeting.
Absolutely. You have secreted proteins, enzymes for digestion, proteins that live in the membrane. They all need to be routed with perfect accuracy.
They need an address label.
Precisely. And that address, or sorting signal, is encoded right in the protein's amino acid sequence itself, or sometimes in the carbohydrate chains attached to it. These signals get recognized by specific receptors on the coats of those budding vesicles.
So that ensures that, say, lysosome parts only get packaged into vesicles headed for the lysosome.
That's it. It guarantees accurate distribution across the entire cellular city.
It's important to remember that this whole intricate traffic pattern, it wasn't just a guess. It was meticulously mapped out. So let's talk about how scientists actually figured this all out, which brings us to the foundational work using autoradiography.
Yeah, tracking radioactive material inside the cell. This classic work was done by Jameson and George Poulad, and they focused on really active cells, pancreatic acinar cells.
Why those?
Because they're just constantly churning out huge amounts of digestive enzymes for secretion. The challenge was, how do you follow just one batch of newly made proteins when the whole factory is running at full tilt all the time?
And they came up with the brilliant pulse chase experiment.
It was genius. The pulse was a really brief incubation with radioactive amino acids, maybe only 3 minutes long.
So just long enough to label whatever was being made in that exact moment.
Exactly. And then the chase involved immediately moving the tissue to a medium with unlabeled amino acids. So the longer the chase, the farther that initial radioactive batch would have traveled from where it was made.
And the results, I mean, if you look at the figures, they show this clear physical wave of movement through the cell. It just confirmed the whole flow of traffic.
It really did. You can see it plain as day. After the 3-minute pulse, the radioactivity, those little black dots on the film, is located exclusively over the endoplasmic reticulum. That's proof that the ER is the site of synthesis.
Okay.
Then, after a 17-minute chase, the label has moved. Now it's concentrated in the Golgi Complex and the little vesicles next to it. And after a really long chase, say 117 minutes, the radioactivity is only in the secretory granules near the cell surface, just starting to be released.
So that one experiment basically defined the entire biosynthetic pathway end to end.
As a sequential integrated unit, it was foundational.
But that radioisotope work, as important as it was, modern cell biology has largely shifted to using fluorescent proteins. The huge advantage here is you can watch protein movement in live cells in real time.
Right. The classic demonstration of this uses a viral protein, the VSVG protein, which researchers tagged with green fluorescent protein, or GFP, so they infect cells, and the cell basically becomes a factory for this glowing fusion protein.
And they use this really cool trick to get all the proteins to move at the same time, so they could get a clear, synchronized visual.
They used a temperature-sensitive mutant of that viral protein. At a high restrictive temperature, say 40 degrees Celsius, the fluorescent protein gets made, but it misfolds slightly and gets trapped in the ER. It just accumulates there.
So the whole cell's ER just lights up green.
Exactly. Then, when the scientists suddenly drop the temperature to a permissive level, say 32 degrees, all that trapped protein can now fold correctly, and it moves out synchronously in this massive wave. You can literally watch the entire fluorescent pool move from the ER to the Golgi in about 10 minutes.
Called a live-action replay of the pathway Pallot and Jameson had traced.
Just decades earlier, yeah. And now, with things like super-resolution microscopy and lattice light sheet microscopy, researchers can watch multiple organelles, peroxisomes, mitochondria, Golgi all interacting dynamically in a single living cell. The technology is just incredible.
Our third approach is totally different. It's about breaking the cell apart, subcellular fractions.
Right. This is about molecular composition. The idea is you homogenize the cells, you break them open, and all those internal membranes fragment and then reseal into these tiny spherical vesicles.
And then you can separate them based on their density.
Yes. This mixed population of vesicles, mainly from the ER and Golgi, is called microsomes. Using density gradient centrifugation, you can separate the rough microsomes, the ones with ribosomes stuck to them, from the smooth ones.
And once they're separated, you can analyze what they're made of.
Exactly. Modern proteomics, using mass spectrometry, can give you a comprehensive molecular portrait of any isolated organelle.
There's a great example of that with phagosomes.
Oh, yeah. Researchers isolated phagosomes. Those are the vesicles formed when a cell eats something, and they found that these supposedly simple structures had over 160 different proteins in them. Many of them had never been known to be involved in ingestion before.
This molecular approach led directly to the fourth method, cell-free systems, which was perfected by Nobel laureates Rashman and Sheckman.
Right. Instead of a whole cell, you just use the isolated parts. Early on, they used rough microsomes and showed that newly made secretory proteins got trapped inside the lumen. That proved the ER membrane was essential for sequestering them away from the cytosol.
But they took it even further.
They did. By incubating artificial lipid vesicles liposomes with purified coat proteins, they could show that the proteins alone could make a vesicle bud in vitro in a test tube.
So they rebuilt the process from scratch.
Reconstituted it, and the strategy allowed them to identify one by one every single molecular component needed for initiation, for bending the membrane, for selecting the cargo, all without the crazy complexity of an intact cell.
Finally, we get to the immense power of mutant phenotypes, especially in yeast.
Yeah, this was Randy Sheckman's lab. They screened yeast for mutants that had an abnormal membrane distribution. They called them sec genes for secretion.
And the results were just so visually clear.
They're beautiful. Since vesicles have to bud from the ER and then fuse with the Golgi, a mutant that blocked vesicle formation at the ER.
Like a Sec12 mutation.
Right. It caused the cell to fill up with these dramatically expanded ER sheets. Everything was backing up.
Yeah.
But a mutant that blocked vesicle fusion with the Golgi, like Sec17, that caused the cell to accumulate just massive numbers of unfused vesicles floating around with nowhere to go.
The aha moment here, which really defined a lot of modern cellular science, is the idea of evolutionary conservation.
Absolutely. The molecular machinery is so deeply conserved that you can do these amazing experiments where you take the mammalian version of a transport protein and use it to cure the genetic defect in a yeast mutant, and it often works perfectly.
So the functions are essentially interchangeable across hundreds of millions of years of evolution?
It's just incredible. And for more complex cells, like mammalian cells, researchers can use RNA interference, or RNAi. They use these small interfering RNAs to basically turn off a specific gene and see what happens.
Like the experiment with the Golgi enzyme.
Yes, they could knock down different genes and watch to see where a fluorescent Golgi enzyme ended up. If they knocked down a key transport protein, the enzyme would get stuck in the ER. It's a way to pinpoint the exact job of that missing protein. Maybe it was part of the COPII code, or a regulator like SAR1 in that critical ER-to-Golgi step.
Okay, let's focus now on that first major compartment, the endoplasmic reticulum, the ER.
Right. This is a massive continuous network of membranes that penetrates pretty much the entire cytoplasm. It defines this internal space, the ER lumen, which is totally separate from the surrounding cytosol.
And we divide it into two main subcompartments, the rough ER and the smooth ER.
The RER is defined by the ribosomes bound to its cytosolic surface, which makes it look rough. The SER lacks ribosomes, so it looks smooth.
And they look structurally different, too. The RER is usually made of these flattened sacs called cisternae, and it's physically continuous with the outer membrane of the nucleus.
Whereas the SER is a highly curved tubular network, it's more like a collection of interconnected pipelines. And that high curvature is actively maintained by these specialized membrane-bending proteins called reticulins.
But even though they look and act different, the membranes themselves are connected.
They are. We know they're continuous because if you label proteins or lipids, you can watch them diffuse freely between the RER and SER regions.
Let's break down the functions of the SER first. It's really extensive in specific cell types, right? Like muscle cells or endocrine cells.
Exactly. It has three major jobs. First is the synthesis of steroid hormones. That's why it's so prominent in, say, the Leydig cells of the testes.
Yeah.
Second is detoxification, which is a huge job for liver cells. This is handled by a large family of enzymes called Cytochrome P450. These enzymes oxidize thousands of different hydrophobic compounds, things like environmental toxins, barbiturates, alcohol, and convert them into more water-soluble hydrophilic forms that can be excreted in the urine.
And there's that surprising fact about detoxification. It's not always a good thing.
It's absolutely a double-edged sword. That enzymatic process can sometimes create a bigger problem. A classic example is benzoapyrene, a compound you find in charred meat. It's relatively harmless on its own, but these very same detoxifying P450 enzymes can convert it into a potent carcinogen.
Wow. And the third major function of the SER.
Calcium sequestration. The SER is a critical storehouse for calcium ions. In muscle cells, where it's called the Cytoplasmic Reticulum, the regulated release of that stored calcium is the key trigger for muscle contraction.
Okay, now for the RER. This is the starting point of the entire biosynthetic pathway. It's the cell's main manufacturing hub.
It is. In highly secretory cells, like the goblet cells that make mucus, you can see this beautiful polarity that sets up the whole traffic flow. The RER and nucleus are at the bottom, the Golgi is in the middle, and the secretory granules are all packed at the top, ready to go.
And the RER is where proteins and lipids are synthesized and where the first carbohydrate chains are added.
Correct. Which brings us to a really fundamental concept, protein synthesis location. About 1/3 of all the proteins our genome encodes are synthesized on RER-bound ribosomes. This process is called co-translational translocation.
And that 1/3 is a critical group of proteins.
Oh, absolutely. It includes all the proteins destined for secretion outside the cell, all the integral membrane proteins, and all the soluble proteins that are going to live inside the lumen of the endomembrane system, the ER, Golgi, lysosomes, and so on.
And the other 2/3, cytosolic proteins, nuclear proteins.
All of those are synthesized on free ribosomes out in the cytosol and then imported into their respective organelles post-translationally after they're fully made.
So what decides where a protein gets made?
It all comes down to the signal hypothesis. The core idea is simple. The site of synthesis is determined by a specific amino acid sequence that emerges first from the ribosome as the protein is being made.
Let's walk through the steps of that co-translational process. It starts with a signal sequence emerging from the ribosome.
Right. It's this short chain of about 6 to 15 hydrophobic non-polar amino acids. And as soon as it emerges, it's recognized by the signal recognition particle, or SRP.
And the SRP does two things, right?
It does. It binds to the signal sequence, and it also temporarily arrests or pauses translation. This is a brilliant checkpoint. It makes sure the protein doesn't start folding in the wrong place before it even gets to the ER membrane.
So the paused SRP ribosome complex then docks with the SRP receptor on the ER membrane.
Exactly. And both the SRP and its receptor are G proteins. The energy from GTP hydrolysis is what triggers the release of the SRP. That allows the ribosome to then associate with the translocon.
Which is the actual protein channel in the membrane.
That's the channel. And we know from high-resolution structures that it's not just an open hole. It's ingeniously designed. The pore is hourglass-shaped, and when it's inactive, it's sealed by a short alpha helix that acts like a plug.
So nothing leaks out of the ER.
Right. It's the binding of that incoming signal sequence that physically displaces the plug, opens the channel, and allows the new polypeptide to start threading through into the lumen, co-translationally.
And once it's inside, the RER lumen is where a whole host of processing steps happen.
First, the initial signal sequence gets clipped off by an enzyme called signal peptidase. Second, oligosacchαρyltransferase adds the core carbohydrate block. Third, molecular chaperones like BP bind to unfolded segments to help the protein fold correctly.
And fourth, the disulfide bonds.
And fourth, protein disulfide isomerase, or PDI, catalyzes the formation of those critical disulfide bonds, which are vital for the stability of proteins that are going to end up outside the cell.
Okay, so what about integral membrane proteins? They use the same translocon, but they obviously don't go all the way through.
Right. Their hydrophobic transmembrane segments are actually shunted laterally from the translocon channel directly into the lipid bilayer. The translocon has a kind of side door or a lateral gate that opens and allows that hydrophobic segment to just dissolve into the lipid core.
What determines which way the protein faces? The final topology.
It's all about charge. There are positively charged amino acid residues that flank the cytosolic end of that transmembrane segment. The inner lining of the translocon is thought to orient the segment, so the more positive end is always left facing the cytosol.
And that first orientation sets the pattern for the rest of the protein.
It does. Every subsequent transmembrane segment then has to flip 180 degrees to maintain that alternating pattern.
What about the newer class of tail-anchored proteins? Their signal sequence is at the C-terminus, the very end. They can't possibly use this co-translational path.
That's right. Their signal only emerges when translation is already finished. So they need a completely different post-translational targeting route called the GET pathway. After they're synthesized, cytosolic proteins grab them and shuttle them to a special complex on the ER that finally inserts them into the membrane.
We have to touch on membrane asymmetry. It's established in the ER and then it's maintained throughout the whole trafficking journey. Why is that so important?
It's fundamental. The two sides of the membrane are functionally distinct. Whichever domain faces the cytosol in the ER will always face the cytosol. And the part that's in the ER lumen will eventually become the external surface of the plasma membrane.
So the ER lumen is biochemically like the outside of the cell.
It's functionally analogous to the extracellular space. It's where you have high calcium, it's where disulfide bonds can form, and it's where those initial carbohydrates are added.
The ER is also the birthplace for most of the cell's lipids. So how does membrane lipid modification work?
Right, most membrane lipids are synthesized there. And because they're usually inserted first into the cytosolic leaflet, the cell needs a way to keep things balanced. Enzymes called flipases actively flip some of these new phospholipids into the opposite leaflet to maintain symmetry.
But if all lipids start in the ER, how do different organelles end up with dramatically different lipid compositions?
There are a few mechanisms. First, enzymes and other organelles can chemically modify the lipid head groups. Second, budding vesicles can preferentially include or exclude certain lipids. And third, and this is increasingly important, are lipid transfer proteins, or LTPs.
The ones that work at those membrane contact sites.
Exactly. They facilitate lipid exchange directly between organelles at those contact sites without any need for membrane fusion.
Let's go back to glycosylation, specifically N-linked glycosylation. This is where that core carbohydrate chain gets built.
Right, and it's built on a lipid carrier called Dolichol Phosphate. The process is invariant in mammalian cells. The cell first constructs this standardized 14-sugar block. It's always 2 N-acetylglucosamine, 9 mannose, and 3 glucose residues.
And then the whole thing gets transferred at once.
The whole block is transferred en bloc by oligosacchαρyltransferase to a specific asparagine residue on the new polypeptide.
And that invariant sugar stamp is basically the passport that allows the protein to enter the ER's rigorous quality control system.
It's the cellular checkpoint. The process starts when 2 of the terminal glucose residues are trimmed off. The glycoprotein, now with 1 glucose left, binds to an ER chaperone like calnexin.
And when that last glucose is removed, the protein is released.
It's released. But here's the key. If the protein is still misfolded, an enzyme called UGGT recognizes the exposed hydrophobic bits and it adds a glucose back on.
So it sends it back into the chaperone cycle for another try. It's a recycling loop.
It is. But this can't go on forever. If folding repeatedly fails, a slow-acting enzyme starts trimming off mannose residues instead. This is the destruction sentence. Once those mannose residues are gone, the protein is permanently prevented from recycling and is marked for degradation.
Wait, so the ER spends all this energy bringing proteins in and folding them, only to spend more energy shoving them back out into the cytosol to be destroyed. Why not just destroy them inside the ER?
That's the critical paradox, and it defines ER-Associated Degradation, or ER. Misfolded proteins are dislocated back out of the ER through the membrane channel and into the cytosol. There, they're deglycosylated and destroyed by these massive molecular machines called proteasomes.
Why do it that way?
We think the cell uses the cytosolic proteasome because it's the central, powerful, all-purpose degradation hub. But it does seem counterintuitive.
And this process can have really serious clinical consequences.
Oh, absolutely. Over 60 human diseases are linked to ERAD errors. The classic example is cystic fibrosis, the most common mutant CFTR protein, which might actually be marginally functional if it could just get to the plasma membrane.
It gets destroyed by the quality control system sting.
Exactly. The ER's QC system deems it fatally misfolded, and it's destroyed prematurely by ERAD before it ever has a chance.
When misfolded proteins start to pile up faster than ERAD can clear them, the cell triggers this massive emergency response, the unfolded protein response, or UPR.
The UPR is monitored by these critical ER sensor proteins like PERK and ATF6. Normally, these sensors are kept quiet by the chaperone BP, but when misfolded proteins accumulate, all the BP gets recruited to deal with the overload.
Which frees up the sensors to activate the response.
It does, and there are two main outcomes. First is the PERK pathway. PERK dimerizes and it phosphorylates a critical translation initiation factor. By doing that, it inhibits the vast majority of new protein synthesis.
It hits the emergency stop button.
It does. It reduces the flow of new cargo into the ER, giving the factory time to clear the existing backlog.
In the second pathway, ATF6.
ATF6 is actually transported to the Golgi Complex. There, its cytosolic domain is cleaved off. This domain then migrates to the nucleus and acts as a transcription factor, stimulating the expression of hundreds of genes designed to fix the problem. More chaperones, more transport proteins, more QC components.
So it's a comprehensive fix-it mechanism. But this response has a really serious existential implication.
It does. If these corrective measures, the shutdown and the fix-it genes, don't work after a period of time, the UPR pivots and triggers a cell death pathway, apoptosis. The decision to survive or self-destruct is literally managed right there in the ER lumen.
So assuming the protein survives all that, the final step is the journey forward, ER-to-Golgi vesicular transport.
Right. Transport vesicles bud from these specialized regions of the ER that lack ribosomes, called ER exit sites, or ERS. These COPI-coated vesicles quickly fuse with each other to form these interconnected structures called the ergic or vesicular tubular carriers, VTCs. And these VTCs then begin their journey towards the Golgi complex, moving along microtubule tracks.
Which brings us to the cellular post office and processing plant, the Golgi complex.
It has a very characteristic organized morphology. It's stacks of these flattened, disc-like cisternae, usually curved like a shallow bowl.
And the Golgi has this clear polarity which defines the processing path.
Yes, it flows from the cis or entry face, which is closest to the ER, through the medial cisternae to the trans or exit face. The two critical sorting hubs are the cis-Golgi network, or CGN, which is like the first quality control checkpoint.
Deciding if protein should be sent back to the ER or move forward.
Right. And then the trans-Golgi network, or TGN, which is the final major sorting station that directs all the traffic to the plasma membrane, lysosomes, or storage granules.
And we can actually see this polarity by looking at where the enzymes are. Different enzymes live in different parts of the stack.
Exactly. Specific mannose trimming enzymes are only in the medial cisternae. The enzymes that do the final sugar additions are only in the trans cisternae. It's a true processing plant where the carbohydrate chains started in the ER are sequentially modified as the protein moves through.
Let's follow that glycosylation in the Golgi. This is where the N-linked modification continues.
As proteins pass through the cis and medial cisternae, most of those original mannose residues from the ER are trimmed off. Then, a whole series of different glycosyltransferases Sequentially add a diverse range of other sugars and acetylglucosamine galactose sialic acid.
And the strict spatial arrangement of those enzymes is what creates all the final diversity in the carbohydrate chains.
That's it. And the Golgi isn't just finishing the ER's work. It's also where O-linked oligosaccharides are built entirely from scratch. And it's the site for synthesizing massive complex polysaccharides like proteoglycans.
Okay, now let's get into what you called arguably the most important conceptual shift in Golgi biology. The debate on movement through the Golgi.
Right. For decades, the leading idea was the vesicular transport model. This model suggested that the cisternae themselves were stable, fixed structures. They were like the rooms in the post office. And cargo was just shuffled from room to room in tiny anterograde vesicles.
But that's not what we think anymore.
The pendulum has swung dramatically. The current consensus is the Cisternal Maturation Model, which says the cisternae themselves are dynamic and transient structures.
So they're not fixed rooms. They're more like moving walkways.
That's a great analogy. They form at the cis face by the fusion of incoming carriers from the ER, and they physically progress or mature into medial and then trans cisternae, changing their enzyme composition as they move.
What's the evidence for this shift in thinking?
There are several key pieces. First, if you block transport from the ER, the Golgi complex literally disappears. And when you restart transport, it rapidly reassembles. That's consistent with the cisternae being short-lived transient structures. Okay. Second, and this is a big one, really large cargo molecules like the huge procollagen complexes secreted by fibroblasts, you can see them staying inside the cisternae as the cisternae themselves move. They're way too big to ever fit into one of those tiny transport vesicles.
So if the cisternae are moving forward and carrying the cargo, what is the role of all those vesicles we see in the Golgi?
In the maturation model, the vesicles aren't primarily carrying cargo forward. Instead, they are functioning to carry the resident Golgi enzymes, like that manosidase, the second enzyme, in a retrograde direction from trans back to cis.
Ah, so that's how a newly forming cisterna gets the right enzymes for its specific stage of processing.
Exactly. It maintains the unique composition of each maturing cisterna as the whole structure moves forward.
Okay, let's discuss the engineering behind this movement. Coated vesicles. The cytosolic surface of these budding membranes is always covered in this fuzzy protein coat.
And that coat has two essential simultaneous functions. First, it's the mechanical device. It literally forces the membrane to curve and physically pinch off to form the vesicle.
And second, it's the selection mechanism.
Right. It provides the adapters that are needed to capture the specific cargo, the right membrane proteins, the soluble cargo, and the docking machinery for that particular trip.
We have three major types. Let's start with COPI-coated vesicles. These handle transport from the ER to the Golgi, the anterograde path.
COPI vesicle formation is regulated by a small G protein called SAR1. When SAR1 is activated by GTP, it inserts an alpha helix right into the cytosolic leaflet of the ER membrane. That physical insertion is what starts the bending of the bilayer.
And that initial bend then recruits the other coat components?
Yes. It recruits the Sec23, Sec24 complex. Sec24 is the primary adapter. It binds directly to the ER export signals in the cytosolic tails of the cargo receptors. And then the rest of the complex, Sec13, Sec31, forms the outer structural cage.
What kind of cargo does COPI carry?
Well, it carries things like glycosyl transferases and docking machinery that the Golgi needs, but also soluble cargo that's bound to receptors. And if you have a problem with one of those cargo receptors, like ER-to-C53, it can lead to some serious genetic disorders.
Like the bleeding disorder.
Right, an inherited bleeding disorder where essential coagulation factors can't be secreted because they can't get out of the ER in a COPI vesicle.
Next up, COPI-coated vesicles. This is the essential retrograde or backward traffic.
COPI vesicles move materials backward from the Golgi back to the ER. And crucially, they're also the ones responsible for carrying the resident Golgi enzymes retrogradely between the maturing cisternae. Their assembly is regulated by a different G protein, ARF1.
The primary function of this COPI retrograde path is retention and retrieval. It's how an organelle maintains its identity when membranes are constantly flowing.
Exactly. Retention is sort of passive. You just exclude big protein complexes from the budding vesicles. But retrieval is active, and it relies on an address label. Soluble ER-resident proteins have a specific retrieval signal, the 4-amino acid sequence KDEL at their C-terminus.
Why do you need a retrieval mechanism like KDEL if those proteins are supposed to stay in the ER anyway?
Because the system is leaky. No sorting mechanism is 100% perfect. KDEL is the safety net. It ensures that any essential ER-resident protein that accidentally escapes in a COPI vesicle gets recognized by the KDEL receptor in the cis-Golgi.
And that receptor then binds to the COPI coat.
And facilitates the active return of the escaped protein right back to the ER where it belongs.
Okay, moving past the Golgi, the TGN is the sorting station for the digestive pathway. Let's look at the highly specific process of sorting lysosomal enzymes.
Right. Lysosomal enzymes get a unique sorting signal right there in the Golgi. Specific enzymes catalyze a two-step addition of a phosphate group to their mannose residues, creating the definitive address, mannose 6-phosphate, or M6P.
And that M6P signal is then recognized by the M6P receptors in the TGN.
The MPRs bind the M6P tagged enzymes, and this whole complex gets incorporated into our third type of vesicle, clathrin-coated vesicles, which bud from the TGN.
And how do the receptors get linked to the clathrin coat?
Through specific adapter proteins like the GGA proteins, they're the physical bridge. They link the cytosolic tail of the MPR, which is holding the cargo, to the outer clathrin network.
Okay.
Once that vesicle delivers its contents to the low pH environment of an endosome, the MPR and the enzyme dissociate. The enzyme goes on to the lysosome, and the MPR gets recycled back to the TGN to do it all over again.
This very specific molecular address system really highlights how devastating the defects can be, which brings us to the clinical context of lysosomal disorders.
Exactly. The discovery of the M6P address actually came from studying a condition called eye cell disease. In these patients, their lysosomes are basically empty. The digestive enzymes are all secreted outside the cell instead.
And the defect was?
A deficiency in the enzyme required to add the M6P tag. Without the M6P address, the cellular post office just doesn't know where to send the enzymes.
And then there are the broader lysosomal storage disorders, or LSDs, where a deficiency in just one of those digestive enzymes causes a toxic buildup of whatever it was supposed to digest.
Right, like Tay-Sachs disease, where the lack of an enzyme leads to a toxic accumulation of a specific lipid in brain neurons. Or Gaucher's disease.
And the treatment for Gaucher's is this brilliant example of using cellular targeting to save lives. It's called enzyme replacement therapy.
It's an elegant solution. The challenge was, how do you get the purified replacement enzyme specifically into the affected cells, which are macrophages? Okay. The successful approach involved chemically treating the purified enzyme to expose some of its underlying mannose residues. It turns out macrophages have mannose receptors on their surface, so they recognize this modified enzyme and efficiently internalize it by endocytosis, delivering it right where it's needed.
So smart. Okay, let's address the final step of logistics. Targeting vesicles to specific compartments. Fusion can't be random.
Right. The first step is initial contact called tethering. The specificity here is conferred by Rabs. These are a huge family of small G proteins. We have over 60 of them. And they basically give each membrane compartment a unique molecular ID tag.
And the Rabs recruit the tethering proteins.
Exactly. The active GTP-bound Rab on the vesicle recruits these long, fibrous proteins or large multi-protein complexes that literally reach out and capture the incoming vesicle.
And then the actual docking and fusion is mediated by the SNAREs.
The crucial components. SNAREs are integral membrane proteins. We have v-SNAREs on the vesicle and t-SNAREs on the target membrane. When the membranes get close, the SNARE motifs from each side interact to form this incredibly stable 4-stranded alpha helical bundle.
And that bundle acts like a molecular zipper, generating immense force.
It does. It zips up and pulls the two lipid bilayers into intimate contact. This mechanism is so essential that it's the target of some of the most potent toxins known. Botulism and tetanus toxins are proteases that specifically cleave neuronal SNAREs, which blocks neurotransmitter release and causes paralysis.
It's a high-stakes failure of that fundamental docking mechanism. So after docking, we have fusion.
The force generated by that SNOR bundle is generally enough to induce the membranes to merge, opening a fusion pore. And once fusion is complete, the SNARE bundle has to be disassembled. That's done by an ATPase called NSF, which uses ATP energy to twist the complex apart, freeing up the components for the next round.
This whole sequence culminates in exocytosis, the fusion of the vesicle with the plasma membrane.
Right. And in regulated secretion, like in neurons, it's incredibly fast. The arrival of a nerve impulse causes a sudden influx of calcium ions. A calcium-binding protein senses this, triggers the final fusion step, the pore dilates, and the contents are rapidly discharged.
Let's briefly look at a newer application of this science. Extracellular vesicles, or EVs, for drug delivery.
This is a really exciting field. EVs are these tiny vesicles that most cells release naturally, and they carry RNA and proteins between cells as a form of communication.
And the idea is to use them as a sort of a natural nanoparticle for drug delivery.
Exactly. The appeal is that they're naturally occurring, so they can move through the body largely unnoticed by the immune system, and they might even be able to cross tough barriers like the blood-brain barrier.
But there are still big challenges.
Huge challenges. Their half-life in the body is really short, and you have to figure out how to load them with your therapeutic cargo and then how to target them to a specific tissue. That usually means engineering their surface to display some kind of tissue-specific membrane protein, giving them a custom delivery address.
Okay, moving on to the cellular demolition crew, lysosomes in animal cells.
These are the digestive organelles. They contain over 50 different acid hydrolysis enzymes that break things down, and they all work optimally at a very low acidic pH of about 4.6.
And that low pH is actively maintained
By a V-type proton ATPase in the membrane. It's constantly pumping hydrogen ions into the lumen to keep it acidic.
How does the lysosome avoid digesting itself?
The internal surface of its membrane is lined by these highly glycosylated proteins. The dense carbohydrate chains form this protective shield that keeps the potent enzymes away from the membrane itself.
So lysosomes break down stuff from the outside, but they also have this crucial role in internal maintenance.
Autophagy.
Autophagy is the regulated destruction and replacement of the cell's own organelles. It's essential for cellular renovation. An organelle that's targeted for destruction gets surrounded by this double membrane structure called a phagophore.
Which then seals up to form an autophagosome.
Exactly. And then that autophagosome fuses with a lysosome, becoming an autolysosome, and that's where the degradation happens. This process is critical for responding to things like nutrient deprivation. The cell literally cannibalizes itself for energy and for clearing out damaged components or invading bacteria.
And after digestion, what's left over is a residual body, which can sometimes be retained indefinitely as a lipofuscin granule. You see these accumulating in long-lived cells like neurons, and they're associated with the aging process.
We should also mention plant cell vacuoles.
Right. In many mature plant cells, a single central vacuole can take up 90% of the cell's volume. Its membrane, the tonoplast, also has a proton pump to keep it acidic.
And plant vacuoles are like a combination digestive tract, storage locker, and pressure regulator.
That's a great way to put it. They share the lysosome's digestive function, but they also handle massive storage of ions, sugars, and sometimes toxic compounds for defense. And most importantly, by pumping in ions, they generate high hydrostatic pressure, or turgor pressure.
Which provides mechanical support for the whole plant.
Exactly. It provides support, and it's the force that stretches the cell wall during growth.
Okay, let's talk about movement in the other direction. The endocytic pathway. This is how cells bring things in.
Right, we can categorize it broadly. There's bulk phase endocytosis or pinocytosis, which is just non-specific fluid uptake. But then there's receptor-mediated endocytosis, or RME, which is selective and incredibly efficient.
RME starts at these specialized regions of the plasma membrane called coated pits.
Right. Coated pits are where specific receptors and their bound ligands like hormones or proteins from the blood get concentrated. The pits then invaginate, pinch off, and form clathrin-coated vesicles.
Let's break down the clathrin-coat structure. What's the basic building block?
The basic unit is called a triskelion. It's made of 3 heavy chains and 3 light chains, all joined at the center. It literally looks like a 3-legged pinwheel.
And these triskelions overlap to form the coat.
They overlap extensively to form this polygonal basketwork, like a geodesic dome made of hexagons and pentagons.
The inner layer of that coat is the adapter. For RME, that's the AP2 adapter complex.
AP2 is essential. It's what binds to the cytosolic tails of the receptors to concentrate the cargo, and it's also what links that inner layer to the outer clathrin cage. The whole process is regulated by specific lipids in the membrane called phosphoinositides, which trigger a shape change in AP2 that allows it to bind the cargo.
And the final pinch-off, the vesicle fission, requires this powerful dynamic protein.
That would be dynamin. It's a large GTP binding protein that polymerizes into this helical collar right around the neck of the invaginated pit. When it hydrolyzes its bound GTP, it undergoes this conformational change, a kind of twisting motion that physically severs the vesicle from the membrane.
The classic story here, of course, is the discovery of LDL cholesterol uptake via RME by Brown and Goldstein.
Right. This all started from studying patients with familial hypercholesterolemia, or FH, who have dangerously high cholesterol. They found that normal cells could bind to LDL, but cells from FH patients couldn't. This led them straight to the LDL receptor.
And the identification of the famous JD mutant really proved the mechanism.
It did. The JD mutant receptor could bind LDL just fine, But because of a single amino acid defect in its cytoplasmic tail, it couldn't get concentrated in the coated pits.
So it couldn't be internalized.
Right. It proved that localization in the pit was the critical step, and that this was governed by specific sorting signals in the receptor tail that are recognized by the AP2 adapter.
So once the vesicle is internalized, the material enters the endocytic pathway for sorting.
Right. It goes to the endosomes, which are like the distribution centers. We distinguish between early endosomes, which are the first sorting station near the cell periphery, and late endosomes, which are maturing as they move closer to the nucleus.
And sorting at the early endosome is crucial.
It's highly selective, and it's determined by the receptor type. Housekeeping receptors, like the LDL receptor, will dissociate from their ligand in the low pH of the endosome. The receptor then gets recycled back to the plasma membrane for reuse, while the ligand moves inward to the late endosome.
But not all receptors get recycled.
No. Signaling receptors, like the EGF receptor, are often marked for destruction. This is a process called receptor downregulation.
And that destruction relies on a specific molecular tag.
A critical tag. These receptors get marked by the enzymatic attachment of a small protein called ubiquitin to their cytosolic tail. These ubiquitinated receptors are then sequestered into these tiny internal vesicles that form inside the late endosome.
So the late endosomes become multivesicular bodies, or MVBs.
Exactly. And this ensures that when the MVB finally matures into a lysosome, the receptor itself gets destroyed along with the ligand.
This whole molecular pathway has huge clinical implications for diseases like atherosclerosis.
It does. Atherosclerosis is basically a chronic inflammation that's initiated by the deposition of oxidized LDL in blood vessel walls. Macrophages eat this LDL and become these bloated foam cells, which contribute to the dangerous plaque formation.
And understanding the LDL receptor pathway led directly to drugs.
To statins, which block cholesterol synthesis, but also to newer drugs like PCSK9 inhibitors, which work by preventing the destruction of existing LDL receptors in the liver. This means the liver can clear more LDL from the bloodstream. It's a direct therapeutic application of understanding that receptor trafficking pathway.
Finally, in this section, we have phagocytosis, the uptake of large particles.
Right. In mammals, this is mostly a protective function carried out by professional phagocytes like macrophages. The engulfment process is driven by the dynamic movements of actin microfilaments. The large particle gets enclosed in a phagosome, which then fuses with a lysosome to become a phagolysosome.
And a battle against pathogens is really won or lost right at this step.
Absolutely. Some bacteria, like Mycobacterium tuberculosis, can hijack the pathway by inhibiting that crucial phagosome-lysosome fusion step. Others, like Listeria, just escape the phagosome entirely by destroying its membrane and living in the cytosol.
Let's shift gears now to the last section. Proteins made on free ribosomes that are imported after synthesis, or post-translationally.
Right. Into three key organelles: peroxisomes, mitochondria, and chloroplasts. These processes still rely on specific signal sequences, but the mechanics are totally different. For peroxisomes, proteins use a peroxisomal targeting signal, or PTS, and are shuttled by a receptor called PX5.
And what's really unique about peroxisomal uptake?
It's the only one of these three systems that can somehow import proteins in their native, fully folded conformation. The other two require the proteins to be straightened out first.
Okay, so for mitochondria, proteins have to cross two membranes.
They do. Most matrix proteins have an N-terminal presequence as their targeting signal. Cytosolic chaperones keep the polypeptide unfolded before it passes through the TOM complex in the outer membrane. Then it engages the TIM complex in the inner membrane.
And what powers the movement across that inner membrane?
That specific step is powered by the electrical potential, the voltage across the inner mitochondrial membrane. Once the polypeptide is partly in the matrix, chaperones pull it the rest of the way in using one of two fascinating mechanisms.
Let's discuss the less obvious one, the Brownian ratchet.
It's such an elegant idea. It's biased diffusion. Instead of an active motor pulling the protein in, chaperones inside the matrix just bind to the polypeptide as soon as it enters the matrix space.
And that binding prevents it from sliding backward.
Exactly. It prevents backward diffusion. So the protein diffuses forward randomly, gets locked in place, diffuses forward a bit more, gets locked again, and so on. It ratchets its way in without a dedicated motor.
And finally, chloroplasts, which are even more complex internally.
Similar principles apply. They use toctic complexes. All chloroplast proteins use an N-terminal transit peptide. But a protein that's destined for the innermost compartment, the thylakoid, has to have an additional signal sequence, the thylakoid transfer domain. It's a second-level molecular address.
Okay, we've covered a huge amount of ground.
We really have. We've completed our deep dive through the cell's entire internal logistics network. We started at the ER assembly line with its quality control, moved through the dynamic maturation of the Golgi, traced all the specialized vesicle routes with COPI and COPII, learned about the precision docking from Rabs and SNAREs, and finally explored the functions of lysosomes and endosomes.
And here's where it gets really interesting, I think. The sheer necessity of traffic balance. We've seen these sophisticated mechanisms for growth, for biosynthesis, and equally sophisticated mechanisms for destruction, like autophagy or receptor destruction.
Right.
The cell's functional integrity depends completely on maintaining these specific traffic patterns. The correct recognition of KDEL, the N6P signal, the internalization addresses in receptor tails.
It's a constant balancing act between growth, damage repair, and recycling, all based on the cell's internal conditions.
And if you think back to the unfolded protein response we discussed,
Yeah.
if those ER stress sensors detect that the factory is just hopelessly overwhelmed, that misfolded proteins can't be cleared, and the resources are exhausted.
The cell's ultimate response isn't just to repair.
No, it's to activate a programmed cell death pathway.
That dynamic, self-destructive decision-making process, managed right down at the molecular level by sensors in an organelle, it just highlights how cellular life is constantly balanced on this molecular knife edge, even in the most fundamental compartments we've just explored.
Thank you for joining us on this deep dive into cytoplasmic membrane systems. We look forward to seeing you on the next deep dive.